Okay, good morning all students. I'm going to start my lecture. With chemiosmosis. Students who are in WebEx can shift to YouTube. Students who are joined with WebEx can join YouTube. Okay, I wait for 4 minutes more. Yesterday we discussed about the electron transport chain. And moving forward with the process, today I am going to discuss chemiosmosis. Students who are live right now, you can comment on YouTube. Okay, if I am audible. So in the first phase of oxidative phosphorylation, the first was the transfer of electrons. Now second phase is of phosphorylation. Second phase is of phosphorylation, in which the production of ATPs occur. Okay, as a result of part 1, oxidation of NADH and FADH2, an electrochemical gradient is created. An electrochemical gradient is created. Okay, meaning there is a difference. There is a difference in the electrical charge between the two sides of the inner mitochondrial membrane, and the outside or the exterior of the mitochondrial membrane is positive because of the accumulation of protons. Okay, so the inside is negative due to the loss of the protons. Okay, so this part yesterday we discussed was electron transport chain. Now moving further, as now the electrochemical gradient is produced. Okay, which leads to formation of a proton gradient in the intermembrane space. Okay, and the inner membrane here. Okay, here number of protons will decrease, and this proton gradient formed will lead to generation of a proton motive force, through which ATP synthase will convert ADP into ATP. Okay, through which it will convert ADP into ATP. Okay, so the theory of chemiosmosis, also known as chemiosmotic hypothesis, was originally given by the scientist Peter Michel. Okay, and is now one of the widely accepted theories in generation of the ATPs. Okay, and the respiratory chain is effectively utilized to produce ATP from ADP, and the concept of chemiosmotic hypothesis is comparable with the energy stored in a battery separated by positive and negative charges. Okay, the inner mitochondrial membrane as such is impermeable to the protons and OH ions. Okay, this transport of electrons through electron transport chain is coupled with the translocation of protons also. Okay, with the translocation of protons also across the inner mitochondrial membrane from the matrix to the intermembrane space. The pumping of electrons results in an electron chemical or proton gradient or a proton gradient. Now this one is outlined simply. Okay, diagrammatic view of the oxidative phosphorylation of the chemiosmotic hypothesis or chemiosmosis. Okay, this one all is an outer mitochondrial membrane, intermembrane space here, and this one is a inner mitochondrial membrane. Okay, in this membrane there will be low concentration of hydrogen ions, whereas in the intermembrane space there will be high concentration of hydrogen ions. Okay, specifically protons. Okay, which will help in the generation of the ATP, which will help in the generation of the ATP. Now how proton gradient works. Diagrammatic presentation here. Outer mitochondrial membrane, intermembrane space, inner mitochondrial membrane, and the mitochondrial matrix. It's a sectional view of the mitochondria. Okay, and given here is a V respiratory chain complexes F0 and F1 protein subunits for the phosphorylation, which are basically subunits of the ATPases. Okay, which are the subunits of the ATPases. After the completion of the electron transport chain, at a time 6 protons are pumped from intermembrane space to the mitochondrial matrix, okay, which leads to generation of a force in a rotary manner of the enzyme ATP synthase, which leads to formation of a ATP, per 6 hydrogen proton ions. 3 ADPs are converted into 3 ATPs. Okay. Now the enzyme system for ATP synthesis. We have already discussed the ATP synthesis, various types of ATP synthesis found in various organisms. Right now I am going to discuss how they work actually. Okay, they work in a rotary type of manner, and this is a diagram of a ATP synthase. Rotary diagram of a ATP synthase. Here intermembrane, and this part is a mitochondrial matrix. This part is a F0 and this one is a F1, and this whole function in a rotary manner. Okay, this whole function in a rotary manner. Okay, now moving further for the functioning, as I told you right now, this ATP synthase function like a rotary motor, okay, or rotary system. Okay, so let us assume that this one basically it's a part at which ADP and phosphate are taken up by the ATP synthase, okay, and this further will be synthesized ATP, okay, and ATP will be released, okay. So at a point at three different categories, ADP and PI will be taken up by the system, okay. ADP and PI will be taken up by the system, and ATPs will be released in the mitochondrial matrix, okay. ATP will be released in the mitochondrial matrix. Now the chemiosmotic mechanism. Electron move in a chain of membrane bound cages spontaneously driven by the high reduction potential of the oxygen and relatively low reduction potentials of the various reduced substrates that undergo oxidation in the mitochondria. So the diagram shown here, okay, is a scenario of a mitochondrial ATP generation. Okay, it's a scenario of a mitochondrial ATP generation. Okay, this one outside of the membrane is an intermembrane space. Electron donors basically, okay, are transferred from various complexes, and the final will be oxygen. Final electron acceptor, or specifically speaking, terminal electron acceptor will be oxygen. You can view clearly that the hydrogen ions will be accumulated at the end of the electron transport chain. Now comes the role of the ATP synthase. The energy of electron flow is stored as a electrochemical potential in the form of protons, and the ATP synthase uses this electrochemical potential to synthesize ATP. So when does it get activated? ATP synthase gets activated. It gets activated when there is a high concentration of hydrogen ions in the intermembrane space, which produces a force in order to activate ATP synthase enzyme. Which converts ADP into ATP. Okay, which converts ATP into ADP. Okay. Now this was a scenario where a mitochondrial membrane is present. Okay. Now I am going to discuss in chloroplast. Okay. Now I am going to discuss in chloroplast. Okay. The electron transport mechanism will be similar to earlier ones, but there will be a slightly difference here. Okay, as the light is a part of the system. So starting with the process, light converts water to a good electron donor. Okay, whereas in the case of mitochondria, we have seen NADH and FADH2 as electron donors. Okay. Now second one, electron carriers pump hydrogen ions in as electrons flow to the NADP plus. Okay, and this energy of electron flow is stored as a electrochemical potential. And this electrochemical potential, which is formed, okay, as you can see clearly in the thylakoid membrane, it is negatively charged. Outside the thylakoid membrane, thylakoid membrane, it is negatively charged. Due to the accumulation of hydrogen ions in the membrane and the force generated through the electrochemical potential, it will activate ATP synthase enzyme, which leads to formation of ADP into ATP, which leads to formation of ADP into ATP. Okay, and this is how the mechanism works in the case of chloroplasts. In both organelles, electron flow creates an electrochemical potential by the transmembrane movement of protons and positive charge. In both the cases, this electrochemical potential drives ATP synthesis by a membrane-bound enzyme ATP synthase. That is fundamentally similar to both the mitochondria and chloroplasts in bacteria and archaea as well. Okay, and as mitochondria is not present in bacteria, yes, it is not present in bacteria. Okay, some of the bacteria are also photosynthetic ones also, which we are going to study tomorrow or day after tomorrow. Okay, how its photosynthetic mechanism works in the absence of chlorophyll, but bacterial cells do contain chlorophyll and popularly known as bacteriochlorophyll, which are different from the plant chlorophyll, but their functions are almost similar to the plant chlorophyll. Okay, so this is an electron tower, okay, which compounds can be better electron donors or better electron acceptors. Okay, electrons move spontaneously from the donors with more reduction potentials to acceptors with more positive reduction potentials. Okay, so as I told you, oxygen is the terminal electron acceptor, so it is the best electron acceptor given here. Okay, it is the best electron acceptor. Okay, and the above ones given here, such as carbon dioxide, hydrogens, are electron donors. They are the best electron donors, and the value given in volts, E0 in volts, is given here for each of the donors also. Okay, so moving downwards, okay, carbon dioxide, hydrogen, carbon dioxide which is converted into methanol, NAD plus which is converted into NADH, carbon dioxide again which is converted with acetate, sulfur which is related with the conversion of hydrogen sulfides, pyruvate which is converted into lactate, FAD, sulfates, fumarates, coenzyme Q, okay, cytochrome C, nitrates, nitrites, okay, for the nitrites conversion into nitrogen, which is a process of particularly denitrification, ferrous ions, and finally oxygen atoms, by identifying which type of compound is involved for the electron donors and acceptors, you can find out whether the organism is aerobic, anaerobic. Okay, you can find out easily. Okay, the ones which use oxygen as a terminal electron acceptor, as I told you in the last lecture also, are basically aerobes. But in the case of facultative anaerobes, they may also use other compounds also. Okay, they may also use other compounds also, which I am going to discuss today. So what happens if we run out of oxygen And how do we break our food? To break energy. Okay, how do we make our food? Break down food to make energy. The body has a plan B also for this situation called fermentation. Okay, called fermentation. It happens all the time in athletes, runners, when they use all their oxygen and produce lactic acid Fermentation starts after glycolysis, replacing citric acid cycle and oxidative phosphorylation. During glycolysis, only 2 ATP molecules are produced. NADH is then oxidized to transform the pyruvates made in glycolysis into lactic acids. We have discussed earlier also regarding this matter. I had given you example of Louis Pasteur, okay, which is who gave a concept of Pasteur concept. Okay, during fermentation, in order to get a particular product of our interest, the level of oxygen is basically decreased for the cells respiring in the fermentation system, which leads to higher production of the products in the fermentation medium. Okay, and now how many ATP do we get per glucose in cellular respiration? We have already discussed this in last lecture, about 30 to 32 ATPs. Okay, 30 to 32 ATPs. Okay, but this may vary according to type of organisms also. Okay, this may vary with the type of organisms also. Okay, it may be prokaryotes or it may be eukaryotes. Okay, why? The reason is very simple. Okay, because in the mitochondrial membrane, proton gradient also needs some power in order to carry out transport of protons. Okay, in order to transport protons. Okay, so there may be some subtle system which helps to transport protons, which helps in a higher generation of ATPs. Okay, so instead some of it must be used to transport molecules into and out of the mitochondrial matrix also. Okay, which is not present in prokaryotes, so they are more energy sufficient. Okay, so they are more energy sufficient. Okay, as compared to eukaryotic cells. Okay, because there are no such transports present in the prokaryotes, as no mitochondrial membrane is present in them. Okay, so in this case ADP must be transported into the matrix, so it can be made into ATP, and ATP must be transported out, so it can be used by the cell. So the 30 to 32 ATP figure accounts for the transport of ATP and ADP, which uses energy from the proton gradient, meaning that there is less energy left to drive ATP synthesis, yielding fewer number of ATPs. This is how the count of ATP molecule was reduced due to this process. Okay, due to this process, and if we take into account all these parameters, okay, the ATP generation or ATP yield will be lower. Okay, will be lower. Okay, in real cells, yield of ATP per glucose molecule would be actually lower than the 30 to 32 ATPs. Okay, actually lower than the 30 to 32 ATPs, and cells often use proton gradients to transport other molecules in addition to the ATPs and ADPs, and some of the proton gradients energy may be also lost if the inner mitochondrial membrane is leaky to the protons Okay, there are some chances if there is a leak, or there may be some damage due to any of the problems also. Okay, there may be a problem. Okay, so 2 net ATPs are made in glycolysis, another 2 are made in the citric acid cycle. Beyond this 4, the remaining all the ATPs come from the oxidative phosphorylation, in which two coupled processes, electron transport chain and chemiosmosis occurs. And based on lot of experimental work, it appears that 4 hydrogen ions must flow back into the matrix through ATP synthesis to power the synthesis of one ATP molecule. Okay, to power the synthesis of one ATP molecule. So for production of just one ATP molecule, okay, 4 hydrogen ions must flow back into the matrix. When electrons from NADH move through transport chain, about 10 hydrogen ions are pumped from the matrix to the intermembrane space, so as each NADH yields about 2.5 ATPs. Okay, yields about 2.5 number of ATPs. Okay, and in the case of FADH2, the drive pumping of only 6 hydrogen ion leads to the production of 1.5 ATPs per FADH2 molecule. Okay, so finally, instead it must hand its electron off to a molecular shuttle system that delivers them through a series of steps to the electron transport chain. And some cells of our body have shuttle system that delivers electrons to the transport chain via FADH2. In this case, only 3 ATP are produced for 2 NADH of glycolysis. Other cells of our body have a subtle system that delivers electrons via NADH, resulting in the production of 5 ATPs. In bacteria, both glycolysis and citric acid cycle happen in the cytoplasm, so no shuttle is needed. Okay, so no shuttle is needed. Thus, they produce 5 ATPs. Okay, so they produce 5 ATPs. Now, I am going to start with the bacterial electron transport chain. TY students, please be present. Okay, this is important for you. Okay, it is somewhat similar to the other electron transport systems also, but what I am going to discuss right now, what is basically a difference between aerobic electron transport chain and anaerobic electron transport chain. Okay, and remember, the different organisms and even E. coli under different conditions will use electron transport systems that employ a variety of substrates and terminal electron acceptors, and thus the respiratory systems can be visualized as being branched at the dehydrogenase and terminal oxidase sites. So why there is a variety? Okay, it because some of the bacteria are autotrophs, some of them are facultative anaerobes, some are pure aerobes, some are obligate anaerobes. So there is a wide variety in the requirement of the bacteria, which leads to use of different substrates, okay, and different terminal acceptors according to the need of the cell. So electron transfer reactions are initiated by the specific modular units. These may be NADH, lactate, succinate, formate, or glycerol 3-phosphate dehydrogenases, okay, which are generally associated with the electron transport of the E. coli. Okay. Okay. Right now I am going to discuss the electron transport chain of the Paracoccus denitrificans, okay, under aerobic conditions. Under aerobic conditions. Okay. Now moving forward, with the Paracoccus denitrificans, okay, you can see the process similar: coenzyme FES complex, coenzyme Q complex, almost similar, cytochrome C FES, cytochrome A3, okay. Okay, but in this system you can clearly see the presence of the enzyme methanol dehydrogenase. Okay, this one is a methanol dehydrogenase. Okay, and this is how the system in bacteria differs with the other electron transport chains. Okay, so the process almost similar. I am repeating the whole process again. Okay, NADH will be converted into NAD, hydrogen ions will be pumped into the periplasm. The electrons are transferred from FES to coenzyme Q to cytochrome B, FES, cytochrome C1. At this point also, hydrogen protons are basically pumped in the periplasm, and these electrons are transferred to the cytochrome C, and cytochrome C will be transferred to the cytochrome A and cytochrome A3, which in a combined manner will act as a oxidases. At the same time, okay, methanol will be converted into aldehyde with the help of enzyme methanol dehydrogenase. Okay, methanol dehydrogenase, and further they are passed on to cytochrome C551 to cytochrome C. Okay, so now I am going to move forward with the type of electron donor modules and electron acceptor modules in the case of aerobic conditions and anaerobic conditions. Okay, these are the expected molecules. There may be also different modules which basically work. This example given here is an modules of the E. coli. Okay, so starting with the first one, hydrogenase enzyme 2, where hydrogen will be converted into hydrogen ion. Okay, the second enzyme is the lactate dehydrogenase, where lactate will be converted into pyruvate. Now third enzyme, succinate dehydrogenase, where succinate will be converted into fumarate. Okay, now the fourth enzyme, alpha glycerophosphate dehydrogenase, only under aerobic conditions right now. Okay, where alpha glycerophosphate will be converted into dihydroxyacetone phosphate. will be converted into dihydroxyacetone phosphate. Next one is the formate dehydrogenase. Next one is the formate dehydrogenase, where formate will be converted into carbon dioxide. Okay, where formate will be converted into carbon dioxide. Okay, and now these electrons will be transferred to Q8. Okay, will be transferred to Q8. What is Q8? It is a ubiquinone. It's an ubiquinone. Okay, and one more dehydrogenase is a NADH dehydrogenase. Okay, under aerobic conditions, okay, or maybe anaerobic conditions also, NADH will be converted into NAD. Okay, NADH will be converted into NAD. Okay, now as electron pass through the Q8, which is a ubiquinone, okay, there are three conditions which are assumed right now. One is the high concentration of oxygen. Second one is the low concentration of oxygen, and third one are the anaerobic conditions, and the third one are the anaerobic conditions. In the conditions of high oxygen concentration, where cytochrome C will be used, which converts oxygen into water, okay, and the second condition, right, which is the condition of low oxygen, where cytochrome D will be used, okay, where oxygen will be converted into again water. Okay, in the case of high oxygen concentration, cytochrome C is used. In low oxygen pattern, cytochrome D will be used, which also converts oxygen into water. But in the conditions of absence of oxygen or anaerobic conditions, enzyme nitrate reductase will be used. Okay, nitrate reductase will be used. Okay, where NO3 will be converted into NO2, that is nitrates to nitrites. But everyone please remember, different types of bacteria use different types of reductases. Okay, according to the type of substrates, according to the type of molecules available. Okay, so the last dehydrogenase enzyme, okay, under the anaerobic conditions, alpha glycerophosphate dehydrogenase under anaerobic conditions converts alpha glycerophosphate into dihydroxyacetone phosphates. But now electrons will be transferred to the MQ8, which is a menacunone. Okay, and menacunone is generally activated under the conditions of anaerobic, which finally, where fumarate will act as a terminal electron acceptor, and fumarate will be converted into succinate with the help of enzyme fumarate reductase. Okay, any questions? If you do not have questions, you can comment no sir, so that I can move forward. You can comment in YouTube itself, students. Okay, I'm moving forward then. Now, as I already discussed this one in the diagram. So some of the electron acceptors used in the respiration. Okay, this will be your table, will be your explanation for the anaerobic respiration also. Okay, so one by one I am going to discuss different types of electron acceptors used in respiration, and most of the part will be of anaerobic respiration, So starting with the first one, which is an example of oxygen for the aerobic respiration. Okay, and finally the product will be water. Okay, and examples are various aerobic bacteria, fungi, protists, etc. Okay, now the anaerobic respiration part, which is the main part of our of the anaerobic respiration, of which first one is the nitrate, which is converted into nitrite. Examples of this process, which is being found in the enteric bacteria, Next one, where nitrites will be converted into NO2 minus, N2O or N2. Okay, this process is basically a denitrification process, and organisms involved are Pseudomonas, Bacillus and Paracoccus. And sulfates will be converted into hydrogen sulfides, H2S. Okay, examples are disulfovibrio and disulfatomaculum. Okay, next one is a carbon dioxide, which will be converted into methane. Okay, this process occurs under anaerobic conditions of all methanogens and acetogens. Okay, these methanogens are those bacteria which produce methane. Next one is where Fe3 plus will be converted into Fe2 plus. Examples of the bacteria are Pseudomonas, Bacillus and Geobacter. Next one is HSO4 minus will be converted into HASO2. Okay, HSO2. And the examples are Bacillus, disulfatomaculum and sulfurospirillum. Next one is SiO4 minus will be converted into directly elemental acid or HSiO3 minus. And the examples of these bacteria are Aeromonas, Bacillus, Thawazia. Okay, next one is fumarate. Okay, which will be converted into succinate. An example of this process is a Vollinella. Okay, the bacteria involved will be Vollinella. And the last one is the S0, which is elemental sulfur, will be converted into hydrogen sulfide. Examples are disulfuromonas and thermoproteus. Okay, now moving forward with the anaerobic system of the same bacteria, which is a Paracoccus, denitrification. Okay, but under anaerobic conditions, not in the aerobic conditions. Okay, the difference you will see here is the conversion of nitrates to nitrites. Okay, and this will be converted into NO and into finally nitrogen oxide to dinitrogen oxide, and finally converted into nitrogen. This whole process is of a denitrification. Okay, is of a denitrification. The process is almost similar. Rest of the process of electron transport chain is almost similar. Okay, is almost similar. Okay, but you can see clearly now here in this case cytochrome A is not present. Okay, cytochrome A is not present here. Okay, instead cytochrome D here will play a major role. Okay, in the conversion of nitrites to the NO, to the NO. Okay, now this is just an example. Okay, in the case of Paracoccus denitrifications, I am explaining the whole process again. NADH will be converted into NAD and passed on to the first complex, and the hydrogens will be pumped into the periplasm. Okay, and these electrons will further move through the coenzyme Q to the cytochrome B and to the cytochrome C1b FES complex. At the same time, NO3 will be converted into NO2 minus, nitrates to nitrites, and now electron will now move forward to the cytochrome C, and from cytochrome C it will move to the cytochrome C and cytochrome D1 complex, where NO2 minus will be converted into NO. At the same time, cytochrome V also transferred to a Cu copper containing system. Okay, which will help in conversion of N2O into N2. At the same time, NO will be converted into further N2O, okay, dinitrogen oxide, and finally N2 and N2O will be converted into nitrogen. And this cytochrome C will be further, electrons will be further transferred to the cytochrome B, cytochrome C and FES complex. Now moving forward, the more of the cytochrome C, this is a diagram, okay, which is basically involves succinate and fumarate. Okay, above example was of a denitrification. Now this example is of a succinate fumarate system. Okay, now starting with the step 1, fumarate succinate will be converted into fumarate. Okay, for in which a succinate dehydrogenase enzyme will be used, and hydrogen atoms are formed, and Q will be converted into quinone will be converted into QH2, will be reduced basically. Okay, now moving for the further system, here ferrous electrons will be transferred to the FES systems, and finally to the cytochrome C as well as cytochrome B. Okay, from B to the lower energy level to the higher energy level, okay, will be further electrons will be transferred from Q, where quinone will be converted into Q minus, and again this Q minus will be used by the system. Okay, now in the step 2, okay, the QH2 transfers the protons into the intermembrane space. Okay, and Q is released, and Q will be converted into Q minus, and Q minus will be converted into QH2 from the 2 hydrogen ions which are present in the matrix, and the whole cycle is again similar to the above one. Okay, so I am going to end my lecture.